Alright, in this video I want to talk a little bit about the Golgi apparatus, its role, and also the role of glycosylation in regulating protein folding and monitoring if the protein is folding correctly. So the Golgi apparatus is basically considered the post office of the cell. This is going to be where all of the all the proteins that go through the endomembrane system that come from the ER are going to be processed. They're going to be coded for destination sorting, and they're going to be packaged into the appropriate vesicles headed for the correct destination, right? So with the exception of those endomembrane proteins that have an ER retention sequence and therefore should stay in the ER, the Golgi is where all the rest of them are processed and sent to the right place. And even those with an ER retention sequence, as we talked about before, often need to be retrieved back to the ER, and so that's happening in the Golgi as well. So part of this processing involves protein folding, so the proteins have to continue to fold in the correct manner along the way. It involves processing the glycosylation, so removing sugar residues and adding new sugar residues, as we'll talk about, and then ultimately getting them into the right vesicle that has the right coat, that has the right helper proteins, and then is headed to fuse with the right destination. And so we talked a lot in the last class about how those vesicles are formed and how they are formed with the right cargo, the appropriate cargo, and with the right signaling molecules so that they fuse with the correct destination. Most of that, not all of it, but most of that is happening in the Golgi. It certainly happens in the ER and a few other places, but the Golgi has to do this really intricately because the Golgi is sending things to many different areas. From the ER, things are mostly going to the Golgi, right? From the endosome, things are either going to the Golgi or the lysosome. But from the Golgi, they're going all over the place. Alright, so when these vesicles come from the ER, they will fuse together and form the cis. The cis is the entry of the Golgi. So the Golgi has several cisternae, and each of these cisternae basically have different functions in terms of processing those proteins. So I have a few things listed here. Most of these are related to glycosylation and adding or removing certain sugars as those glycosylation carbohydrate residues become mature and become more complete along the way. But we go from cis to medial to trans, and the trans is where things are sent out. All right, so things enter the cis. This would be like your freshman year of high school or college. Then you get to your sophomore, kind of junior years, and then you're ready to be sent off after your senior year. So if you think of this kind of as the learning center and the processing center for proteins, as they enter their freshmen, they continue to mature a little bit more, sent on until they're seniors, and then they are ready to go on and do their jobs. So let's take a look at these vesicles as they move. The center here, the green is stained for some Golgi, and the center is very bright green because that is the main part of the Golgi. And so in order to be able to see these vesicles as they move, the imaging here becomes overly saturated so that we can see those vesicles along the way.
Proteins exiting the Golgi apparatus on the way to the cell surface are often packaged into tubular transport vesicles of significant size. Such tubular vesicles can branch and fragment before they fuse with the plasma membrane. The transport vesicles move along microtubules, which are stained here with a red fluorescent dye.
So you can see that, again, now we see the Golgi as yellow, but that's because it's on microtubules. So microtubules are stained red, Golgi is stained green, and when you have the green fluorescence at pretty much the same spot as the red fluorescence, that makes it look yellow. So we can see these vesicles moving along the microtubules. And we know this, we actually saw this in the very first video on the first day of class, where we saw our fun little motor protein, the kinesin, walking along the microtubule, pulling the huge vesicle behind it. So we'll learn in Unit 5 all about those motor proteins in the microtubule highway system a little bit more. But basically, you can see here the Golgi budding off from the trans and sending those vesicles towards the plasma membrane where they're supposed to go. All right, so let's talk a little bit more about glycosylation. And so the original glycosylation actually happens in the ER. So we're going to take a step back from the Golgi and just talk about the addition of these oligosaccharide sugar residues to these proteins. But my first question for you is, on which side of the ER phospholipid leaflet does glycosylation occur? So we have the cytosolic side, and then you have the ER lumen. So where is that glycosylation going to occur? Well, this out here would be our cytosolic region, and this would be the ER lumen. And we know that glycosylation only occurs inside the ER lumen. Why? Because we know that the inside of the ER is the same as the outside of the cell. And we learned in Unit 2 that glycosylation only happens on the extracellular portion of our lipids and our proteins. So the cytosolic portion are not glycosylated, the extracellular regions are. And since the interior of the ER and eventually the interior of the Golgi is the same as the extracellular of the cell, because remember those vesicles are going to bud, fuse, and release everything inside the vesicle to the outside of the cell, that means the inside of the ER is where glycosylation is going to occur, because the inside of the ER, same as outside of the cell. And so this glycosylation, one major type at least, is called N-linked glycosylation. N-linked because it happens on asparagine, whose single letter amino acid abbreviation is N. And then the glycosylation occurs because this dolichol is a lipid. It's a type of lipid that's in the phospholipid bilayer. It is glycosylated. It has this huge oligosaccharide set of sugars bound to it, right? And these sugars include N-acetylglucosamines, a whole bunch of mannoses, and then these glucoses. And as the protein is inserted into the ER, whether it's a membrane protein or a protein that's going to be secreted that's fully into the ER, as it is inserted, this asparagine will pass by. And there's an oligosaccharyl transferase that basically recognizes if this asparagine is supposed to become glycosylated. Now, it's believed that there are kind of certain sequences of amino acids before and after the asparagine that this recognizes. There might even be a little bit of fold already. It's really not completely clear how this oligosaccharyl transferase knows which asparagine is supposed to be glycosylated or not. It does appear to be fairly well regulated, although there are differences even with the same proteins at times. So this is one of those mechanisms we don't know a lot about. But we do know that not all asparagines are glycosylated. So this is going to see, oh, this asparagine needs to be glycosylated. It will take this oligosaccharide from the dolichol lipid and basically transfer it onto our asparagine. And then once it's on the asparagine, it actually is going to become processed a little bit. And so again, just a quick reminder that the glycosylation happens on the inside of the lumen of the ER. All right. Now, eventually, and this is part of the processing we're going to talk about in the Golgi, eventually this will get trimmed. This is the base of all N-linked glycosylation. Even though they all start with this huge oligosaccharide attached, they are going to be trimmed. These glucoses will be taken off. A lot of these mannoses will be taken off. And eventually, it's trimmed down to this. And then from there, new and different sugars are added. And this is what makes all of the oligosaccharides very different on each protein. So each protein, once it's trimmed down to here, will have different types of sugars in different branches. And so all those oligosaccharides will look very different in the end. But this is part of that processing, the trimming of the original large oligosaccharide, and then ultimately the addition of the appropriate sugars to this base. And so this glycosylation kind of trimming and ultimate re-addition of other sugars, this is a really important aspect of making sure the protein is folding correctly along the way. So if we go back to our school analogy, this is kind of the way of monitoring if that protein is learning correctly along the way before you send it on to the next step. So in this case, is the protein folding appropriately? Is it getting stuck in an incorrect fold, or is it slow to fold? You want to make sure it's at the right step before you move it on to the next part of the Golgi and so forth. So how does that work? Well, those enzymes that are in the different parts of the Golgi, so you have certain enzymes that are in the cis, certain enzymes in the medial, and certain enzymes in the trans, they have different jobs in the processing. So even before this leaves the ER, all of these glucose residues will be taken off. And if those glucose residues aren't taken off, it will not leave the ER and go to the Golgi. So it basically is the loss of these glucose residues that allow this to bind to the receptor that puts this protein in the vesicle on the way to the Golgi. So if it hasn't been processed yet, it won't move forward. And then in the cis, you continue to see some trimming, and then towards the medial and trans, so in the medial you continue to see trimming, and then in the trans you start to add very specific ones. And some of the additions of these are going to be really important for tagging it to go to certain regions. So take-home message here, as the protein folds, the attached oligosaccharides are either trimmed early on, and that trimming is required to get to the next stage. So basically it's like your exit exam. If you haven't been trimmed fully yet, you're not going to move on to the Golgi, or from the cis Golgi to the medial. You're not moving up to the next grade, essentially. And then ultimately the sugars become modified and new ones added, and that kind of helps the fully folded and mature protein be able to get where it's supposed to go and then have its final function. Alright, so again, if the protein is not folded yet, the processing will fail to continue, or the processing will even revert one step. So how does this work? Well, think about it. There's enzymes that are required to remove these glucose residues. Or over here in the cis, there's enzymes required to remove these residues, or to add these residues. Those enzymes will not recognize the protein if it's not folded to a certain extent. Even those enzymes have to recognize a lot of different types of proteins, and therefore are kind of promiscuous in that they recognize a multitude of folds. There are very common misfolded traps that proteins get in, and that enzyme will not recognize that misfolded trap, and therefore that processing won't happen. That protein will not move forward. And in some cases you can actually be reverted back a step. All right, so let's think about this for a second. So we've got our new N-linked oligosaccharide that still has its glucoses here. So a big part of that is to get onto the Golgi is to remove these glucoses. So there's enzymes, this one's called calnexin, that removes that sugar, that glucose, and once the glucose is removed, now this is able to bind to the receptor, get in a vesicle, and head to the Golgi. However, there are other proteins in here that can recognize the misfold, and if they recognize the misfold, they will actually add that glucose back on, basically reverting this a step so that it's not ready to move to the Golgi. So the key take-home message here, the processing of the oligosaccharides help monitor the protein fold, prevent it from going to the next step in the ER, cis, medial, or trans Golgi, prevent it from going to the next step too early, and sometimes even revert it back one so that it has a chance to try and refold better. So just like a school system where you cannot move on to the next grade unless you've learned all that you need to learn, and sometimes you can even be reverted back, although I think that's a little bit harsh in the protein, but not only did you not pass 4th grade, but maybe you need to go back to 3rd grade. But you can relearn that stuff, and these proteins, therefore, by the time they are ready to be released from the trans Golgi, they are folded correctly, they're going to have the right function, and they are hopefully going to do the correct job. And that's a huge way of preventing disease, because we know that misfolded proteins can cause disease. All right. Again, this is important enough that I'm going to just kind of give that final rules. The glycosylation helps promote proper folding, makes sure they're folded properly, and marks the progression. So make sure the protein is folding appropriately to move forward through the Golgi. The glycosylation at the end, so once it's fully glycosylated, they have some really important roles because that's protection. That's actually going to protect from degradation and proteases. So if protein is covered in these sugars, proteases don't cleave it as well. And this is, for example, the glycocoats on bacteria that help protect them from a lot of things. And our intestinal lumen cells, the extracellular proteins are highly glycosylated because that protects those proteins from the acid and kind of the harsh aspect of our digestive system as well. These can also be really important for specific binding. So just like a protein's fold is really important to what it can bind to and what its functions are, well, that glycosylation might help recognize the protein it's supposed to bind to. So a ligand might only recognize the receptor if it's glycosylated appropriately. So we talk a lot about protein fold, protein function, but the sugar residues that get modified and then added are just as important with those functions. And along the way, they're going to make sure that the protein's ready to keep moving forward. All right, and then finally, we're going to learn all about this for the lysosomal proteins. Sometimes this modification in the end is the tag that is the address that helps it get to the right destination, a la lysosomal sorting. All right? So that protein folding is so important that there's this whole glycosylation process to make sure it's folding correctly before it moves on, and the cell is actually going to do some pretty amazing things with misfolded proteins. So if a cell has gone backwards, right, so it's had that sugar residue cleaved off and put back on over and over again, but it's still not folding correctly, the cell will actually go to extraordinary lengths and release that protein from the ER. This is the only time a protein inside the endomembrane system will ever get out. But when it gets out, it's already tagged for degradation by ubiquitination, and it will immediately be degraded. So if you fail a certain grade too many times, basically this protein is eliminated. All right, the last thing I want to talk about with the Golgi is two models of transport. So this is one of those things where there's still a little bit of a controversy, and there's actually some evidence for both of these as to how this occurs. So the vesicular transport model, so this is actually something you do need to know. It will be on the exam for sure. So will a lot of the other things, but I'm just giving you a heads up on this one. The vesicular transport model basically says that the cis, medial, and trans, things move from there through vesicles. So in other words, the cis, medial, and trans are static. They're staying the same, but the proteins move forward through vesicles. So to go from the cis to the trans, you get loaded in a vesicle, and you bond to the next one. Then when you're ready to move forward to the next grade, you bond to the next one, and the next one, and the next one, and then you're sent out. Right? This is kind of like the school system, where the 10th grade teacher has the same class every year, but the students move to the teacher. So the students moved from the freshman class to the sophomore class, and then we'll move from the sophomore class to a brand new class in the 11th grade, and so forth. The classes stay the same, but the students move. And the students can move forward, and the students can move backwards if they're demoted a grade in the ways we just talked about. So we have forward transport and retrograde transport. But the key thing here is that everything moves in vesicles, the cisternae stay the same. This is different from the cisternal maturation model. So in this model, all your vesicles from the ER fuse together, and this actually becomes the new cis. Then the new cis matures together and becomes the new medial, and the new medial eventually becomes the trans, and then this will fully bud off and break off. And so basically this is almost like a conveyor belt where this cis matures to the medial while a new cis comes in, and eventually becomes the tran and gotten rid of, and there's constantly new cis coming in and so forth. This would be like if the students that came in stayed in the exact same class, but the teachers moved backwards. So here, those enzymes that belong in the medial, for example, when this matures towards a trans, that medial enzyme has to be sent backwards to what's the new medial. And the cis enzyme has to be sent back to what's the new cis. So over here in the vesicular transport model, the students move, and the teachers in the class stays the same. Here, the class stays the same and matures, and the teachers move back to get the new class. Right? And so there's some important advantages and disadvantages of each. So the advantage of this one here is that it is much more energetically favorable. So you're not having to use all the energy to make all the vesicles to move those proteins forward. Instead, the proteins are just maturing. You're just moving the teachers back. So think about it. The class might have one teacher for every 40 students. And so if you're just moving the teacher backwards and the 40 students are staying the same, that's much more energetically favorable. You need a lot less vesicles. Right? Whereas here, you need vesicles not only to move the students forward, but sometimes even move the students back. So this one is more energetically favorable. The other thing that's important is this actually allows for huge protein complexes to move forward. So think about it, a big part of regulating and processing the proteins is starting to have their quaternary structure come together, complexes come together. And so things like collagen are huge, and it's always been wondered how collagen can even get put in one of these vesicles to move forward. So if the whole cisternae is just maturing, it doesn't have to be put in a vesicle to move forward, and these transvesicles, some of them can be pretty huge. Right? So two advantages to this one: more energetically efficient, fewer vesicles, and large things make it through. The disadvantage to this one, the cisternae maturation model, and therefore the advantage to the vesicular transport model, is that here in the cisternae maturation model, all of the proteins have to mature at the same rate. So you're basically waiting on the slowest proteins to mature. It's like the no student left behind rule. You can't send the ones forward when they're ready. You really can't fully stop the other ones before they're ready to go forward. So everything has to kind of mature at the same time. Whereas here, as soon as a protein's ready, it can move forward. If a protein wasn't ready, it can move back. This kind of allows all the proteins to mature at their own rate, if you will. So that's the big advantage there. Which one is true? Well, it depends on who you ask, but I would say both are. I'd say there's a combination. My opinion based on the data is that most of it is the cisternal maturation model. This is true. I think there's a lot of evidence that this happens, but I think there are some vesicles that can move forward so that you can take some of those fast maturing proteins and move them ahead and move the slower ones behind. So I think it's a little bit of a combination of both, maybe mostly the cisternae maturation model. But either way, you need to know the differences between each and the advantages and disadvantages of each.